2, 1. Well, hello everybody. With me I have Dr. James Lyons Weiler, and I know everyone out there is concerned about the this new vaccine that it looks like it's coming out, and President Trump is saying he's going to administer it with with the military, and that it's effective, and then he's had a version of it. Dr. Weiler, can you tell the audience a little bit first about yourself? I mean, why would you be an expert in this field? I mean, why do you know what you're talking about in this area?
Sure. Well, I've been in biomedical research for about 20 years. Expert in genetics, genomics, proteomics, biomarker development for the outcomes, health outcomes under different treatments. I've written a book on Ebola. I've written a book on cures versus profits. I've written a book on autism after reading 2,000 studies. You know, I'm a beast when it comes to reading and researching literature, and not synthesizing but reflecting. My approach to literature is to really take it in and then reflect it, and then what comes out comes out. But in terms of the viruses, I'm a hardcore, tried and true evolutionary biologist. I understand virology. I understand immunology due to my research experience. I've written chapters on immunology. So I'm pretty widely educated. I can't say that I know everything, but nobody knows everything. There's a lot of open questions. And so that's what science is for. And so as a scientist, I would hope that we would try to generate some scientific knowledge as we go along and incorporate that, what we think is scientific knowledge, into our public health policies and medical practices. I'm the CEO and president of the Institute for Pure and Applied Knowledge, where we do research in the public interest without any profit motive.
So we've been dealing with this coronavirus for a while. I mean, they said we're going to do a 2-week shutdown, and then it's all going to, and then we can move on, and everything will be back to business again. And obviously, I believe we passed the 2 weeks. 2-week mark by now, I would assume, but we're still dealing with mass quarantine, businesses shut down. I mean, where's the status of are we still dealing with a threat, or is this just a perceived threat?
I looked at the statistics weekly. Right now, I think we're at the lowest point ever. In at least 40 weeks in terms of the number of deaths that are attributed to coronavirus. And this is still when they're practicing, if you have the infection and you die, then they're going to say it's a coronavirus death. That was started back in March. And so, no, I think we're well beyond the cause for the type of concern that we were originally all worried about and we're talking about. And I had done some projections, some mathematical projections. I made it clear when I did my simulations, this is not about this virus. This is about, I wanted to teach the public about what it's like to model, and how initial assumptions of models can be sensitive, and then what some of the options might be. And through that modeling exercise, which you can go back and watch it on Unbreaking Science, that podcast that I do, you can see that I say, okay, let's say if you have a lockdown, an initial lockdown of say 2, maybe 3 weeks. I modeled it. And let's say if you add in antivirals, and let's say they're 60% effective. Okay, I model that. And let's say if you have some kind of a treatment, like hydroxychloroquine. I mean, I was doing this back in February and March.
Yeah.
Then what I said was we're gonna be able to get back, open up society again. So at that time I was for an initial, really widespread 2 or 3 week vacation, I called it. Everybody's got a national vacation for 2 or 3 weeks, right? Let's get to know your family again, learn the guitar, do something you always wanted to do. But those people who had been possibly exposed through tracking and testing, and those people who were at risk should be given antivirals and treatment prophylactically. This was way back then. So in terms of, the original question of why should people know, look at the crystal ball that I apparently had back then. I kind of knew what I was talking about then, but the most important thing that I learned there was the characteristics of this virus, the demographic, the modeling characteristics, the R naught and the rate of transmission and so on, told me that this was going to be a low burn for a long time. This was, yeah, when you cut this thing out, it has just the right kind of intermediate transmission where it transmits, fast enough to be around for a long time, and that's unusual for coronavirus. And so, I made the prediction that it's going to be a long kind of permanent burn way back then. And all the time I was saying, this has nothing to do with coronavirus, kids. This is just, we're just trying to see what modeling is like. Okay, because I've never wanted perpetual lockdown. I was never for that. I was for an intelligent multimodal strategy, right? And so then it became, well, wait a minute, we're going to teach the public a lesson here. This is awful. Look at how bad it is. And what had happened, of course, was that the CDC mangled the testing. They absolutely mangled the testing. They had a deadly testing program.
And because initially there were so many contaminated tests, and now it seems like the tests that we are using, the PCR test, that you shove the stick up your nose and do the PCR. It seems like they've kind of heightened the sensitivity of that test to a point where you'll detect coronavirus on pretty much anything.
Well, sensitivity is one thing, but the real concern here is false positives. If you're just talking about in general PCR testing, yes, you want as sensitive a test as possible, but then you should confirm what you've amplified. If you just use PCR and you say this virus is present, and you have non-specific amplification of off-target sequences, then you're doing a lot of harm by saying false positive if the consequences of false positive is 14 to 10 days off. Okay, so the CDC actually updated their recommendations that you should only be 10 days quarantine if you test positive. So yeah, I criticized them again way back in March. I said CDC completely, if they gets bad, it's because CDC shipped out flawed tests. They shipped it out with one of the primer reaction pairs, they're called primers, one of the PCR reaction pairs of nucleotides that were incorrect. And so the out there in the field, in the hospitals or wherever the people were testing, they were saying, wait a minute, this kit's really not working. This person clearly has all the diagnostics. So we developed our own PCR test. Our PCR test works, yours doesn't. What's going on? And then they said, okay, well, the problem is sensitivity. No, the problem is that they had the problem of sensitivity and specificity, where they had false positives. If you just have a test that's too sensitive, the outcome is you lock down some people at the beginning, right? You lock down and do contact tracing on too many people, but you still lock down everybody. That's not what happened. What happened was they let a lot of people go off of the Princess cruise ship, and they wanted to let them go in a mall in the city there. And they said no, so they went to a military base. They left the military base, they went to their homes, they came down with COVID. Those are false positives. They were all screened. So the problem from the beginning has been false positives, okay? And the cost of the false positive, the shutdowns of businesses, the loss of the businesses permanently, the unemployment, the deaths of despair, the suicides, all because we think that it's a much larger outbreak than it is because of false positives. The reports of testing fruit and coming back with a positive for coronavirus. What is that? So they said they're not going to say it's a fundamental flaw of the test. They're going to say it's contamination. Yeah, we had some contamination. I don't believe it. So there's no reason in the production of this kit why you would ever have contamination, ever.
Okay, so you feel the test in itself is wrong. It's not that it's contaminated. So hence we should not really believe in the test because it's wrong.
Well, you used a very important word there. I'm a scientist. You used the word feel. And I want to say, I think, okay, as close as a scientist might know, I know, because I was a peer reviewer on Dr. Singhan Lee's study. Dr. Singhan Lee is a molecular diagnostic expert in Milford, Connecticut. And he's been very, very critical of the HPV. He wrote an open letter to the World Health Organization. So I knew this guy was objective. And when I got this paper to review, the journal contacted me and said that we're in a fight with one of the reviewers because the reviewers just stopped replying to us. It turned out that that reviewer had a conflict of interest. He had his own test he was developing. But what Dr. Sinhan Lee did, and you can also catch the entire interview I interviewed him after the peer review was done and the paper was published on Unbreaking Science. He actually asked for and received what are called reference samples. When you get reference samples, when you're developing a new molecular diagnostic kit, you're told what they are. These are COVID-19 positive. So he had a collection of COVID-19 positive and a collection of COVID-19 negative reference samples. He used his PCR kit, and like 40% of the ones that were labeled positive, he couldn't get to amplify with primers that he knew worked on positive samples. And 30% of the samples that were labeled negative, he managed to get amplification. Now you would say, okay, wait a minute, he's getting positives and negatives, who's right? Dr. Lee insists that, well, and this is what the paper says and shows, that the FDA accepts what's called Sanger sequencing, which is the actual determination of the nucleotide sequence that you get from the PCR reaction. Usually from the PCR product, you have a collection of amplified nucleotide sequences. You sequence them, and is it human or is it viral? So you get the actual look at the sequence. None of these kits that are out there actually have that as part of the protocol. None of them have been validated to determine if they're amplifying on target or off target. And so we don't know how all these positives, cadaver test positive, we don't know whether it's an off-target amplification of coronavirus, another coronavirus, or something in the human genome. And so he published this paper and he made the news, and you would think that they would say, okay, wait a minute, the FDA, we have emergency use authorizations for these kits that have not been tested. Now we're going to require testing. So they did. They said to everybody, okay, everybody go test yours. We need to know the sensitivity. You need at least 99% sensitivity. They sent out sensitivity requirements, and the data came back, and the CDC issued a report, the FDA, sorry, issued a report that said, oh, look, all these tests are great, 99% sensitive. I wrote to them and I said, where's the specificity? You never asked them for specificity. You've wasted a golden opportunity here. They were actually about to validate. Why didn't you ask for specificity? I can't understand it. In all the years that I ever did molecular diagnostics, I've never heard of anyone that looked at any diagnostic test and only looked at specificity. It doesn't make any sense at all. Because let's imagine, why don't we all get CT scans every year? Or every month for that matter, for cancer? Why don't we all do that? The false positives.
Yeah.
We'll get so many false positives, we'll all be poked full of holes by like pin cushions for the biopsies. And biopsies come with a risk of infection, so they refuse to do it. It's unethical. But hey, this is just a molecular test, no big deal. Okay, we're going to shut you and your business down. We're going to shut down a whole school. We're going to shut down a city. We're going to shut down a state. Those are major consequences of the cost of the false positives.
Well, I mean, that's the thing. That's the thing is that a normal Joe, I mean, what you're talking about now, a normal Joe, they survive, or they just follow the one-liners that they're told, Fox News or whatever news they're listening to, and that's all they're going by. I mean, yeah, even though, and it's almost like an exercise showing how people don't want to kind of go deeper into what's really going on. I mean,
that's part of it.
Yeah,
that's part of it. The other part of it is that the CDC culture is show the public that we know the truth.
Yeah,
and that there's no ambiguity. And even before I became what I call vaccine risk aware, in my Ebola book, I was very frustrated with CDC. CDC, Tom Frieden would not say to any senator or congressman, frankly, Senator, we don't know. They're afraid to say that. I think the path to trust for agencies, regulatory agencies, would be to show the public what they know and what they really don't know. A transparent, honest assessment about what's really going on. Otherwise, they're never going to get the trust back. And I don't want them to get the trust back. I actually have a plan that got me censored from LinkedIn. The plan is to dismantle CDC, just be done with it.
Yeah.
And create a network of organizations that are funded directly by the Senate. And the specific structure of this entity would be 80 research laboratories who are focused on bio threats and public health threats, regardless of whether it comes from a foreign land or a virus or medicine, allopathy, whatever the threat is to public health. Identify those and write a report. All they're doing is doing research and writing a report. They send, there's 80 reports that are written every year about the prioritized threats. They don't talk to each other. They're working in isolation. And then they send those to the all of the Senate committees. So every year the Senate committee gets 80 reports to go through.
And then that way you have unbiased truth, and then also you have checks and balances, so you don't have just information being funneled through one organization. There's
exactly. Yeah. It's completely crazy that we have all of our bio threat geographically located in one place where the enemy could take it out and then launch, right? So the structure is those 80, they send it to every Senate every Senate committee, and that those committee write a report reflecting agriculture, reflecting whatever the committee is, okay? And then those reports go to the Senate Intelligence Committee. The Senate Intelligence Committee then writes a report informing the president what's going to happen. This depoliticizes everything. It should never be public health should never be a political issue. I wrote this up and LinkedIn censored me. They kicked me off. I had 16,000 followers because I want to decentralize public health with a much more, and by the way, the structure that I just described to you is the very definition of a neural network. Yeah. So if we encoded on the agencies how they're organized, instead of a hierarchy, a neural network, it would by definition be intelligent, okay? So yeah, I had 16,000 followers on LinkedIn. I published this and LinkedIn kicked me off and then had the gall afterwards to contact me. I didn't ask them. They contacted me and they said, by the way, if you'd like to know why we kicked you off, it's because you write articles like this. I never asked them. Isn't that something? Now, who owns LinkedIn? Microsoft owns LinkedIn. Okay, very strange. Yeah. So, I have a good number of articles there that I can't get to, including ones that show that CDC has been cooking the data on influenza deaths for years, since 2014. And they combine the deaths from influenza, coronavirus, SV, RSV into one, and they call it influenza disease. And then by the time it gets into the public's ears, it's influenza. And why? Because there's only 5,000 influenza deaths a year in the United States on average.
And they need to make it a pandemic so that they can
need to make it above 6%. Exactly. Yeah. So when you look at the magic number, you'll see that as the total number of deaths, it's amazing. Just today I looked at it, the total number of deaths actually fluctuates from all causes. There's a sinusoidal type wave in the CDC's data. The number of deaths attributed to coronavirus fluctuates precisely at 6 to 7%, right along there, week after week, month after month. Now, it's like the kid that ate the cookie and he's got chocolate all over his face, right? And like, no, Mom, I didn't eat the cookie. I didn't eat that cookie. It's crazy that they think that they could do this stuff and get away with it. And people call me conspiracy. I don't care. The founding fathers were a conspiracy, so they'll call me conspiracy nut. I don't care. Name calling has nothing to do with science.
Yeah, if you look at the science, and then also, so they were kind of cooking the data. They were bringing then the influenza with pneumonia and SARS and all these kind of things, and now they're wanting
not SARS, just any coronavirus.
Yeah, and any coronavirus.
Not counting SARS and SARS-CoV-2. It was pneumonia, influenza, RSV, SV, and coronavirus, any old coronavirus. So in other words, they're like, if it seems like flu, we'll call it flu, good enough.
So the issue then this year is that they're trying to then separate, the coronavirus or the COVID-19. So they're trying to undo what they did in the past and still be able to prove that it's a pandemic.
Exactly. So what I did was I asked the community to get together and to start FOIAing the data from the states that was being sent to CDC, and FOIAing CDC for the data the final report. So like in March, we started this, scared the crap out of them. They didn't know what to do. So they started falling all over themselves. So like, well, our data aren't right. So you see these massive adjustments. Remember the massive adjustments? oh, it's really not this, it's this. It's like, okay, well, how are they going to, my question was, how are they going to cook the data so that both influenza and coronavirus made more than 6%? That's 12% of total deaths. How are you going to do that? The answer is, well, there's going to be a second surge. It's the twindemic. It's going to be worse than ever. We're going to tell people not to get the flu vaccine this year because it might not be so bad. So that there'll be more cases from flu, so there'll be more deaths. That's exactly what they're doing.
Yeah, yeah.
And it's very, very draconian. There's no compassion there for. They want the numbers, because then they got the flu. Then they have the impetus for the vaccine program.
So so that would mean that if you add the COVID-19 and the influenza this year, it should look pretty much the same as just the influenza the other years.
Let me go on the record here, and I want to say the coronavirus this COVID-19 probably does add to a higher death rate overall.
Yes.
Right. I think it is a serious thing that most people over 70 have a high risk. Thank goodness that kids don't die like they die from influenza. They're basically, their intrinsic immune system takes care of the virus quite handily. This time, we don't know what it's going to be like next season. That's the thing. Watch. I'm going to make a prediction right now that we're going to start seeing an age shift distribution, shifting in the age distribution of deaths because of pathogenic priming. The kids that were exposed this year might turn out to have some problems if they get a coronavirus 19 next year. So, it sounds like I'm advocating for eradication of the disease through a vaccine, and I'm not going there. What I'm saying is just watch for it. Watch for it. This is not a pussycat, virus. It's a serious one. But like every other coronavirus that has come through, well, the only reason why it's going to reach so many people and make so many people sick is because if you flatten the curve instead of I said in March, we need to truncate the curve, not flatten it, because if you flatten the curve, you're going to have perpetual infection. You're going to kill more elderly. You have a perpetual active transmission through flattening the curve. It's the stupidest thing that anybody could ever do with this virus. They needed to follow my protocol, my program, lock down 2 weeks, protect those people. Sweden turned out to do that. Sweden's doing okay. They're criticized for, oh my gosh, look at all the elderly people that they killed, but they're no longer killing their elderly. We're still killing our elderly, if anyone's dying at all. So the other thing is that they're not subtracting the baseline death rate for the age groups. So they say if you're over 70, they don't do actuarial science. They need to hire an actuarial scientist that would just look at it and say, on the insurance data from 2018, 2019, you had a 2.3% risk of dying if you were age 70 or whatever it is. They need to subtract that from the overall death rate from coronavirus. Mm-hmm. Because those people belong to the general population as well. They may have died. Those are the ones that probably died with, not from, but they won't do it. So that'll bring their percentages down by quite a large percentage. So
and that's the thing, is that they're looking at 94% of all the people that died had comorbid conditions. So you don't, they probably would have died anyway within a month or so.
Yeah, that's right. So instead of vaccination being the #1 public health policy, I'm totally serious about this. I think our #1 public health policy should be to find the people that have Th2 skew in their immune system, where they don't have a Th1, Th2 balance.
And what is Th1, Th2 for the listeners?
So that you have T helper cells that are really essential for how the immune system responds to pathogens. They're incredibly important. And Th1 is considered to be, if you have a vaccine and you get a Th1 response, people get really excited. That's rare. Usually a vaccine, especially if it's aluminum adjuvanted, will have a Th2 response. Th2 is more pro-inflammatory, whereas Th1, well, now you're going down the path where you get the memory B cells and you got the right kinds of T cells. The Th2, you're going to end up with cytokines, a cytokine profile that's pro-inflammatory. Th1, you're going to get a cytokine profile that is a healthy, robust response. But in reality, you need a little bit of both for a good. And this thing about the immune system, we think we can fool it. We think we can say, hey, immune system, look, here's a pathogen and a bunch of aluminum, and look, this is going to mimic, an infection. It doesn't. The immune system's too smart. It says, no, this is something like a piece of wood that's stuck in my arm. I have to liquidate the flesh. I have to, so you can pull the wood out, right? It's really gross, but that's what it does, right? We've got to get a lot of amoeboid-like cells in there to chew up the tissue. You've got a bad inflammation. Remember, when people feel sick, you know this very well, you're a naturopath. When people feel sick, it's not the virus or the pathogen that is like going around punching up beating up their cells, right? The cells just, most of the time, unless you have a virus that causes cell lysis, a smart virus will just exit the cell. Thank you for your DNA, for the use of your genome. It's like checking out of a hotel. They checked in the hotel, right? They made up their bed. It's all good. Now we're going to leave. But the illness that we have, the disease that we experience, is actually the immune system overdoing its job or doing its job. So sometimes it's like, if there's a bad cold going around, you kind of feel sniffly. I look forward to a fever because then I know I'm on my way to getting over it. Not a big deal. A little bit of time off, but the Th1 and Th2 balance is what we need. And so the number one public health priority that we should all be demanding is, well, I don't want to hear another word about another vaccine. I want to hear public health priority number one is to find the people who have Th2 skew. They have a more pro-inflammatory, pro-autoimmune immune system, and find out what they need through individualized medicine. Straightforward individualized medicine, one by one, to balance it. So you have more of a Th1 and Th2 balance. And how do you do that? You look at their diet, you look at their lifestyle, you look at their exercise, you look at whether or not they, are they over-vaccinated? Do they experience from hyper-vaccination? And are they just chock full of aluminum? And what can we do about that? I'm I've been very upset with Francis Collins, the director of the NIH, for a long time, right? He should have overseen a massive program in response to the injection of mercury and aluminum to dealing with the chronic illness that those cause. Instead, he joined the denialists, and he now is out on YouTube playing the guitar, singing "Poof, the Magic Virus." This is what he's doing. Seriously, look it up. It's like, it's incredible. Is that a confession on him to write a song and sing a song that it's a magic virus? And that he sings how you have to stay locked down until there's a vaccine. I mean, it's awful. It's just, I'm a musician. It's just awful. It's awful and wrong in so many ways.
Well, I mean, and that's, so kind of going back to the Th1, Th2, I mean, you have the issue, you're talking about priming, disease priming, which obviously happens if you kind of activate or you inject individual with a vaccine, so you're more prone to Th2 reaction, where the immune system is more inflammatory, and you will then have more of the cytokine storm. But then also we're talking about antibody response, and we're talking about that the antibody response will kind of fade away after a few months, but then you have the T cells that have kind of T memory cells, and they still persist. They've done studies on that. So if by doing the vaccine, you're then bypassing that memory, those memory cells.
Yeah, but they knew this. They knew this about coronaviruses for decades and decades and decades. In the 1950s, they used to, they knew this in the 1950s. They used to take coronaviruses and they would try to make a vaccine for coronavirus and then do the challenge test where they would infect people. They stopped doing that. They started moving to animals, and the animals got sick too. So they've known this. It's like they reinvented the immune system. Oh, that's allopathic medicine for you. It's a new semester. We're going to wipe everything on the whiteboard off. We're going to forget everything you ever learned. Not going to transfer any knowledge from this to that. Okay, so no, I mean, we knew this, and the T cells are, of course, you have memory. The mammals have an adaptive immune system with memory, is what they've discovered. I mean, they've known this forever and ever and ever. But the balancing of the Th1 and the Th2 as a public health initiative would, of course, highlight the natural question: what's causing the Th2 skew? What's causing the Th2 skew? What could cause pathogenic priming? It would be a highly vaccinated population that has too much aluminum. You put a new virus from the wild or wherever it came from in them, and it has these characteristics, then that goes away, they get better. Next time it comes around again, they're still full of aluminum, they get a hyper response. They've learned autoimmunity against their own proteins. Their own body creates antibodies that are cross-reactive to the protein in the virus and in their bodies. And I have a paper on pathogen priming. It's a study. It was the first study that came out and said these are the targets. If people are going to get really sick from the viral infection, these are the tissues in which they're going to get sick. And it like 7% of them, 21% of them were immune system protein. There's matches between the virus and immune, like, that's serious. But they're all over the place. There's proteins that are ubiquitously expressed throughout the body. So, now we're going to say everything that's analgia, everything arthralgia, all these mysterious diseases, all these things that we have unknown origin. Oh, it's coronavirus.
Yeah.
Because everybody's going to have coronavirus. So it's not HPV vaccine injury, it's coronavirus.
Yeah.
It wasn't the MMR, it was coronavirus. It's an infection. So they have their way out from all vaccine injuries that are these diffuse, hard to diagnose autoimmune conditions. It's like they did with the chronic Lyme. So unless we can have this conversation and get it opening where we can go, wait a minute, we're causing this. We're causing it all. We're putting too much aluminum in our kids. And we have IPAC has a 3 peer-reviewed publications now, where we developed the pediatric dose limit for aluminum, because the FDA never published a pediatric dose limit for aluminum. And then we showed in the last one, which was just published actually in the Journal of Trace Elements of Medicine Biology, that kids in their first year of life on the CDC schedule are in aluminum toxicity 100% of their days. We used to think it was 70%, but that's before we corrected and adjusted for renal development, the kidneys. So now that we've adjusted for renal development of the kidneys, our estimate is 100% of their days they're in aluminum toxicity. And these are really super important years. And many of these kids are coming out of a pregnancy where they got the Tdap and they got the flu vaccine too. So the 100% serious to change the public health priority from vaccination as a solution to looking at Th2 skew, a population with Th2, Th1 balance would be better able to deal with coronavirus. There'd be a lot less pathogenic priming.
And I mean, the issue, and what people don't understand, I mean, you inject the virus along with the adjuvant aluminum, and you're thinking that just heightens the immune system response to that specific virus. I mean, the truth is that the aluminum just heightens the immune system reaction. So if you have a weak thyroid or weak joint or weak kidney or whatever is weak, there's going to be a heightened immune system response to that tissue because 100%. You have parts of that tissue that's floating around in the bloodstream. And so the immune system is going to have a more sensitive reaction to that. So now you get autoimmunity.
100%. And viral infections last 2 or 3 days, usually in humans, maybe 4 or 5, the typical childhood illness, you might have a prodromal period, but the actual high viremia period is not very long. Aluminum sticks around for 5 or 6 months doing its job. The viral proteins actually adhere to the aluminum. That's its job, to keep it around long enough. It's overdone. And then the other thing is we've got these kids that are getting, and adults are getting aluminum-containing vaccines in a world where there's other viruses. So if you get injected for 5 or 6 or 7 vaccines on a makeup day and you're a kid, and you walk into a pathogen, and that pathogen, I hope it's a virus that you walk into, because if it's a spirochete or if it's a bacterium, they're so much more closely related to humans. Their proteins are very, very similar, similar enough that you're going to get some autoimmunity just because you're carrying so much aluminum in your system. I mean, they really haven't thought this out at all, or they don't care. Molecular mimicry between the pathogen protein and the human protein guarantees autoimmunity if you have an overactivated immune system and it's a prolonged overactivation. They're not kidding anybody anymore. It's over. The game is over for aluminum. Our publication of the pediatric dose limit alone in 2018 totally made the FDA look foolish that they've never done that. And then we're actually calculating, okay, here's the first vaccine, hepatitis B, and then it clears out, and we've done all this modeling. They've never done any dose escalation studies on aluminum adjuvants in animals to try to see what a safe dose is in adult animals or in mice. In fact, the safety the safety science that they cited, the ATSDR, which is an agency in the FDA or CDC that was headed up by Julie Gerberding. She was the director of the ATSDR. She did such a good job burying aluminum and toxicity. They made her the director of the CDC, and then they gave her a job and millions of dollars of shares of stock over at Merck. They used oral types of aluminum in adult mice to assess the safety for injected doses, a different form in infant humans. The oral, right?
Yeah.
Injected. Oh, sorry. Oral versus injected. The mode of delivery. Oral versus injected form, the actual chemical formula, the actual compound itself. Adult versus children. Adult versus infant, and then mice versus humans. That's 4 confounders. And CDC makes its living off of adjusting for confounders to over-adjust to make associations go away when they do dissociation studies. So they've got 4 confounders there, and the FDA says, yeah, that looks good, close enough. Yeah. In fact, the FDA wrote a paper, the MITKIS paper. They said, okay, listen, if you take the aluminum that's coming in, and we make the assumption that, yeah, it's oral, but let's assume it's like this if it's injected. There's 3 body compartments that aluminum goes into, so we'll divide by 3, and the aluminum sticks around a while, so we'll divide the toxicity by 365 days. That was the MITKIS study. Look, it's not toxic. So it's totally not science. There's no data at all. What they should have done is they should have taken infant mice, and I'm sorry to all the animal lovers out there. I'm an animal lover, but the FDA is doing this all the time with other adjuvants. Yeah. They should have taken infant mice and injected the infant mice with doses scaled to body weight that matched the schedule, the whole schedule, not just a single vaccine, the whole schedule. And so IPAC actually invited a proposal by Dr. Chris Shaw, the University of British Columbia, and to do that study to IPAC. So he wrote a proposal, submitted to IPAC, and we're fundraising right now to try to get, so that we're funding, the IPAC will become an extra governmental funding agency on the public's behalf. If we're successful, we'll fund a study, if we reach our goal, we'll fund a study where the infant mice are actually given dose escalation as the FDA should have. And that's I took the proposal, I put it out for peer review, I got peer review comments back, standard stuff. I've done this all the time. And then the IPAC board voted that yes, we can go ahead and try to fundraise for it. And everybody can do a fundraiser for it. If you have a health freedom organization, it's called the Community Aluminum Study. And if we get hundreds of health freedom organizations to raise the funds and either ship the funds to IPAC or send them directly to UBC, it doesn't matter to me. Yeah. Then Chris Shaw can get this science done and published, and then that'll be a really big black eye on the FDA if he finds a problem. Let's be scientific about it. Just doing the study in the first place is a black eye because the FDA should have done it. They're doing. There's a video out there of how they're doing dose escalation studies on mice with other adjuvants, but everything except for aluminum. They won't touch aluminum.
Yeah, it's fascinating, and I would assume then, measuring the liver, the kidney, the neurological. I mean, it'd be fascinating to see the buildup and the impact of aluminum in those different tissues.
Yeah, I seriously have my doubts that the mice will survive because it's body weight adjusted, but it's a lot of aluminum.
Yeah,
it's a lot of aluminum.
So going to, now we're looking at a vaccine that is coming out for this COVID, allegedly President Trump, took it. So what are your thoughts about what the type of vaccine that they are developing? Is it something we've seen before? Is it following the same guidelines as we developed vaccines in the past? How is it feasible that something like that can be out on the market this quickly?
Okay, so to answer the first question, there's a variety of types of vaccines that are being developed. There's the mRNA vaccine from Moderna, okay? And Moderna's vaccine, people are a little bit confused about how it works, but the mRNA technology is to inject an mRNA into the cell, where the ribosomes in the cytoplasm start making the viral proteins. They don't make the full virus, just the viral protein. So then that cell is going to fill up with viral protein and probably die because it's just got a lot of protein it's not supposed to have, okay? And then when it dies, those proteins are released out into the interstitium where the immune system says, what the heck is this? And so you get an immune reaction. The mRNA does not stick around. It doesn't, should not, could not, no reason to expect it's going to enter the genome. There are people that are confused about that. DNA vaccines has a different story, but mRNA vaccines, unproven technology. They never brought one to market. And yet the way that Moderna got their FDA or emergency use authorization was by arguing that it was proven that was existing technology. And it's not. There's no vaccine out of the market at all. Did they follow the same protocols for vaccine safety science as before? I have to say, from what I've seen, unfortunately, yes. The vaccine safety studies that were done in the past are very poor assessors of vaccine safety. They think of efficacy first. You know, the initial studies you're supposed to do for safety is to look at animal studies, and they skip the animal studies to look for pathogenic priming. They skipped over it, and so because Moderna was allowed to skip over it, all the other vaccines are allowed to skip over it. And so they literally have to experiment on us, the public, to see if there's pathogenic priming. Unprecedented, where they have every past study for every other coronavirus vaccine, including SARS, including MERS, showed a problem in the mice after you vaccinate them and then expose them to the natural virus. Now, they could have done those animal studies first. In fact, they could have done those animal studies probably 7 or 8 times over by now, since they were given the permission not to. There's no reason why we don't have those data. There's no valid reason, I should say. The reason that they don't have the data probably because it didn't go in their favor. There's probably a problem with pathogenic priming. You probably have heard about it also as disease enhancement or immune enhancement. But pathogenic priming, and this is why, I think there's probably 14 other studies now on coronavirus that use the phrase or at least cite the paper of pathogenic priming. It puts the liability for the ultimate outcome of autoimmunity and disease, in disease enhancement, on the act of the first exposure. And that's why we're calling it pathogenic priming. Pathogenic priming is the act of exposing the immune system to the protein the first time, right? So then you have whatever happens, and then you have antibody-dependent enhancement. But it's not. It could be antibody. It could also be that you got T cell overreactivity. It really depends. There might be autoimmunity in the immune system itself that stops the T cells from being shut down. There could be a feedback loop. So, that's one of our hypotheses. So we're not really sure what the outcome is of the nature, but those are downstream outcomes, the consequence of willingly and intentionally exposing every person in the population to the viral proteins, knowing that a certain percentage of them will experience pathogenic priming. It's just pure evil. I'm a scientist. I can't say anything else other than it's just pure evil. Over what? Something that has 2,000 deaths a week or something now, where we're down to practically nothing. And so, with all due respect to those people who are dying and their families, look at the number of car accidents. Look at all the other number of causes of death. We don't shut down society for these things. Death has been a part of life since the beginning of life. It's just we're never going to defeat death by locking down society. There's other deaths. There's deaths of despair. There's people killing themselves over the loss of their jobs and the loss of their ways of making a living and the loss of their businesses. And so it's very serious.
It almost seems like this pathogenic priming is that you're creating like a time bomb within an individual. So they may not have a reaction to the vaccine at the time, but then anytime, like you're saying, they're exposed to that type of virus, that is when you're then having a possibly even lethal reaction at that time. So
right, and we make it perfectly clear when we talk about it in the study that it doesn't matter whether you get the virus first or the vaccine. You're still being exposed to, if I had a preference, I would want immunity from SARS-CoV-2 from the virus, because I'm a reasonably healthy person. I would probably get a low viremia, and my immune system would be balanced in its approach to the. So I get the vaccine, I would probably have a stronger vaccine reaction. But if I get the vaccine first, then I'm going to have potential of high viremia on the other side because I'm vaccinated, and then that's where the real problem is. And I think there's going to be such an outrageous uproar where people are, saying, "Wow, the coronavirus is so much worse." Then they'll say there's a mutation that made it more deadly. It's just a mutation. As an evolutionary biologist, I'm offended that they would use evolution as their excuse. You can't hide behind the skirt of evolution. That doesn't mean there's not mutations, but it's amazing that public health officials can turn mutations on and turn them off whenever they want. There was a Yale epidemiologist in Connecticut. I was listening to her testify. I was there live, and she said, one thing that the public doesn't understand is that the measles virus does not mutate. What are you talking about? It's a virus. Of course it mutates. The MMR was invented or put on the market in 1960s, right? Like 1963, and they haven't changed the type of mumps that's in there since then. No. And so the mumps version, and this was my testimony in Connecticut. The mumps version has been mutating every year in the virus and in the wild, and also in the lab. It's mutating, so it's evolving away from each other, and you get antigenic shift. I mean, for a Yale epidemiologist, a faculty member at Yale University, to testify that there's no mutations in a virus.
But however, there's a mutation then and the coronavirus.
If it's useful, we'll say there's a mutation. I mean, it's incredible. Yeah, they have such control over the virus.
They get skills. So I'm curious, is this vaccine that they're developing, the mRNA vaccine, why has it never gotten out on the market? You were saying that this is kind of like old technology. There has to be a reason why it's never been used until now.
Well, from what I can see, everybody else is, really super critical of this disruptive technology. MMRs doesn't have, Merck doesn't own the patent. That's why. You're trying to break into a new market. These would be the hot new guys, because they can update the benefit of the mRNA vaccine is you can update it every year. If there is a mutation, you can update it. They haven't updated the MMR since 1963.
Yeah.
So what does that tell you? They have to go back to the FDA. They didn't build into the process with the FDA, right? That, okay, here's this MMR vaccine. We're going to need to update it every year. Just understand this, FDA. And the FDA would have said, okay, that's the technology. It's got to be updated every year. But they said, no, if we update it every year, then anybody could update it. They could compete with us. And it's all of these market maneuvers.
Yeah.
That's not science. That's not public health. And so when you have the FDA approving of something, and then they would have to do a total resubmit. They would have to do new science on it every year. Well, I don't know how Moderna is going to do it other than just going to waiver on that, because it's a perpetual emergency use authorization. So I think that the reason why I couldn't come to market is because it was a closed market. And that, we need free and open market so that companies get feedback on the quality of their product in comparison, not in collusion to others in the market.
And are there other vaccines that are in the running, or is that pretty much a one that has kind of, because I thought there was like 5 companies that were
there's 5 or 6 front runners, and they all have their phase 2, 3 trials. The way that it's supposed to go is you do the animal trials first, and then if it passes animal trials, then you get approval for phase 1. And phase 1 would be like a dosage variance trial where you're just going to change the dose of the vaccine. They all should have done that. So they did that, and they go, look, we got a strong antibody response. And then they say, well, 100%, some of them said 100% of our patients actually had serious adverse reactions, but that's okay. It's still a success. Maybe 4 out of 10 had a problem in another one. And so then you go to phase 2. Well, what they decided to do was skip the animal studies, misinterpret phase 1, and then combine phase 2 and 3. So now they're just going to do this large, 80,000, 50,000, doses, put them out there and see what happens. and it's risky in terms of investors. It's all exciting. Wow, we've got this new technology and everybody's going to get it. It's a vaccine market, yada, yada. This is risky as hell because what if 25% of those people end up, dying in 3 months because coronavirus is still circulating and they're re-exposed to it, or getting really, really, really sick, like they're terribly, terribly infected. And well, they say, well, we're going to follow it for 5 days. We're going to follow it for 5 weeks, maybe 5 weeks, for safety. They need to do total health outcome awareness for 24 months on these people. So we really know that for 2 years, the effect of this vaccine before they release it. But no, we're just going to accelerate it, Operation Warp Speed. We're going to put it out there. We're going to vaccinate everybody. And I haven't heard for months for months and months. I heard once something about the Trump thing. He's going to use the military. I think he's talking about, just to, I don't know, I'm guessing, but probably just for distribution. I can't imagine that they're going to force vaccinate everybody in the United States. They're going to have a problem in their hands if they do.
Yeah, I think it's for distribution. At least I'm hoping it's for distribution. Yeah. And that people still have the choice. And I'm not sure if they're going to the place that, yes, you have the choice to get the vaccine, but if you don't get the vaccine, we have these safe zones, that you're not able to go into, if you don't have the vaccine. Maybe you can't be on a sports event or go to school or go, I don't know what the steps are that they,
I have no idea. I mean, the fact that the Supreme Courts in a couple of states are shutting down governors on overreach for public health is good. It's also very important that we all stand up. You're going to see something. It's going to be posted online. State violence to enforce public health is un-American. And we've seen the mom in Ohio who was tased. We've seen people arrested for singing at church worshiping, church worshipers singing while they're worshiping for not wearing a mask. We see the Ohio mom was tased for not wearing a mask. The person that was helping the cop arrest her didn't have a mask, if you look at the pictures, but that's another story. Around the world, there are people that are being arrested for just posting on Facebook, come out to the protest, in Australia and so on. The state violence incites vigilante violence. There was an MD that choked a teenage girl for not social distancing, tried to strangle her. He was charged with strangulation, allegedly. And sadly, this morning I read of a case up in upstate New York where a 60-year-old man and an 80-year-old man got into an altercation. The 60-year-old man was trying to cause everyone in a restaurant to wear masks. I mean, he was going after the waiters, the waitresses, he was going after the bar staff. And this 80-year-old man was trying to remind him that we live in a civilized society. And the 60-year-old man pushed the 80-year-old man down and he hit his head and he died. So now we have incited vigilante violence over these draconian public health. This is the mainstream media put the fear into people. The government's showing by example that it's okay, right, to, by all means necessary, this kind of baloney. You're going to see that my plan for this is to have everybody in all 50 states, everybody contact your state representatives, the congressional representative and the senators, and tell them that you want a House resolution and a Senate resolution condemning state violence in the enforcement of public health. That's a very practical thing. It's much better than burning down houses and businesses and protesting and going in the street and yelling your fool head off. Call today your congressional representative. Call them tonight, leave them a message and say that you demand a House resolution condemning state violence in the enforcement of public health policies. There's no reason for it. It's not as though this is Ebola. Okay, it's not as though we don't have examples around the country like Sweden, where if you protect the elderly, you're going to get herd immunity, and now you're not killing the elderly anymore. Okay, why lock down everybody? You don't quarantine the healthy, you quarantine the sick, right? So whoever your congressional representative, if you've never ever contacted your congressional representative, just go to the web and find it in your state. Contact them and plead with them, ask them, demand, whatever you feel comfortable with, that you need a House and Senate resolution condemning use of state violence. This way, and contact your sheriff's office too. Leah Wilson, look up Leah Wilson. They have a contact your sheriffs. Those are elected officials, and ask them, what are you going to do if I show up at a place without a mask? Are you going to arrest me? What are you going to do if I refuse a vaccine? Are you going to arrest me then too? Are you going to force vaccinate me? Are you going to force vaccinate me? Look at my face, tell me in my eyes, you're going to do that to me. They're elected people, the sheriffs are.
Yeah.
But I think what we want to do is both contact your sheriffs and contact your Senate and your senator and your congressional representative. Because if the local police and the state police know that they don't have the backing of the legislature, and that the legislature might sanction, put new rules out restricting them. This is not defund the police. This is just don't use violence.
Yeah.
Don't arrest people over public health. Don't confront them. Don't tase them. Don't baton them. You know, I'm not talking about protesting. If you're a protester and you throw a Molotov cocktail, you're going down. The police are going to take you down.
Yeah.
Right? You know, if you're out there and you're at a football game, you're walking your dog, and a policeman comes up to you and you don't have a mask on, or somebody demands your vaccine papers, the minute we demand our vaccine papers, then that's a totally different let me see your papers, please. That's a very different situation, and I'll change my tone at that point in time. Right now, these are elected officials whose seats really, really matter to them. Yeah. Call them now and let them know that their seats are on the line for over this issue of the use of state violence. And watch for it. Watch my Facebook page for the for those links. There's a collection of links that I'm going to put out about that.
Awesome. Well, I think that's an excellent idea. I mean, I think that's really powerful, and that will cut out a lot of fear within the public, and also, like you're saying, this vigilante actions that are taking place. You know, where people are feeling justified to go after somebody because they're not wearing a mask, and feeling justified and inflicting harm on that other individual. So,
yep.
Yeah. Well, Dr. Lyons Weiler, I'm thank you so much for spending time with me, and this information is just crucial, and we just got to get it out more and more and more. So,
well, thank you. You can find that. You'll be able to find that information and more information also at Millions Against Mandates, MAMM.org. There's a collection of links there that will include this eventually. And you can sign up for regular issuances of notices on these types of issues at MAMM.org, Millions Against Medical Mandates. And if you want to support what I'm doing, if you don't mind if I go into
absolutely,
a little bit.
I would love to.
Okay, so IPAC is a not-for-profit organization, and we're registered in Pennsylvania as a not-for-profit, but your donation is not tax deductible yet. We're still working on that, but it's ipa-knowledge.org. And also, if you catch my podcast on Breaking Science on YouTube or Facebook and you like that, you can support it directly at patreon.com/WWDNYK. That stands for What We Do Not Yet Know. That's the name of the studio that does my podcast. It's my studio. And watch for it. We're going to be doing four-person panel podcasts coming up. They're going to be a lot of fun and very important ones. And if you go to my blog, jameslyonsweiler.com, I've had over 1.1 million readers now on my articles. There's a spot there where you can buy me a cup of coffee, send $5 or whatever. You know, I through these three things, I managed to do everything that I do. And I want to thank you very much, Michael, for having me.
Well, thank you. I so appreciate everything you're doing, and I highly encourage the audience to look you up. And I mean, I read your articles, your research papers, and they're amazing. So thank you so much.
Thank you. We have a lot more coming. So
yeah, thank you.